And gene regulatory networks in evolution and development. Our next speaker is Professor Ellen Rothenberg. She's the Albert Bildung Rudolph Professor of Biology in the Division of Biology and Biological Engineering at the California Institute of Technology, and her talk is titled Causality and Validation in Single-Cell Genomics, Analysis of a Major Hematopoietic Transition. Let's welcome Professor Rothenberg.
Thank you very much. The first thing I want to say is just how enormously grateful and delighted I am to be here, and thank you so much to Uta for organizing this meeting and this whole series of meetings, which are just so inspiring, and it's a great honor to be included. Today, I'm going to take you through some data, and I hope that the path that we travel will essentially show you why we have so much respect for causality. And as I would say, causality is an asymptote. It's not a destination. We basically seek a full understanding of causality. We know that cells are caused to transform their properties in a very predictable way in biology, and we know a lot of the genes that are important for that. We would like to understand how that works, and this represents our attempt to get at that problem, at the answers to that question. So the system that we're studying to try to get at this is one in which multipotent progenitor cells, which start out as hematopoietic stem cells, pick a fate. And the cells start out as being able to give rise to at least a dozen very divergent cell types. They end up fixing on one of these basic cell types, which can be further subdiversified. But the properties that the cells acquire in this process are extremely durably acquired. The cells will keep them through countless cell divisions in the future, countless migratory sites in the body, and that is the development of hematopoietic stem cells to become T cells. Now, normally we define a cell's identity by what genes it's expressing right now, and the transcriptomics that I'm going to show you is all based on instantaneous measurements of what a cell happens to be doing in terms of gene expression. But the real question I want to get at is, how is potential regulated? That is, what is the difference between a cell that in the future can give rise to macrophages, B cells, erythrocytes, and so forth, and a cell which right now cannot do any of those things? And so the question is, how do we get at this issue of these genomic states that are permissive versus non-permissive for future gene expression? And one of the very, very good systems for studying this turns out to be the system of early T cell development, because the progenitors that are going to become T cells migrate from the bone marrow through the blood to the thymus, throughout at least most of early life, and through much of adult life also if you're a mouse, less of life if you're a human. And when they arrive in the thymus, they're away from all the cells that are going to give rise to other hematopoietic cell types, but they are still intrinsically capable of giving rise to many other things. And you can assay that capability at the single cell level. And we know that the cells are going through something like 10 to a dozen cell divisions in the early stages after they arrive in the thymus, but they're still multipotent. They can give rise to dendritic and myeloid cells, especially, and they start out being able to give rise to B cells as well when they first get into the thymus. During the first 10 or a dozen cell divisions, they keep these alternative potentials. They're meanwhile being bombarded by signaling from the microenvironment, and this is most importantly signaling through the notch system. So this is the same notch that drives many, many decisions in other kinds of development. In hematopoiesis, strong notch signaling drives the cells to become T cells. I didn't mean to do that yet. And the point is that after the cells cross a certain transition, but not before, this cumulative experience results in stripping the cells of their abilities to give rise to other things and forcing them only to become T cells. And from that point on, the cells will rearrange their T cell receptor genes. They will become parts of the immune system. All of immunology exists after this. We're going to study these early developmental stages and really what makes the difference between a multipotent progenitor cell and a cell immediately after that that's no longer multipotent. Now, we know that this is a rather large regulatory event. So for many years, people have been able to do cell transfer experiments to show that the cells change potential. They lose the ability to give rise to other things. As long as they stay in the thymus, they will be pushed into the T cell pathway. But up until this point here, removing them from the thymus will allow them to become other things. Beyond this point, they cannot anymore. And what happens in part at this time, we know is a large change in the pattern of accessible chromatin in the genome. And this is just a graphic from one of the papers that our lab collaborated with Keiji Zhao's lab at NIH on to do a genome-wide atlas of what was changing in the genome. And these are DNA accessible elements that decrease in accessibility. These are elements that increase in accessibility, which usually means regulatory activity as the cells are going forward. And of all of the elements that change their status between hematopoietic stem cells and these definitive T cell precursors, there's this hot spot for change right in here. And that coincides to the same transition that we're talking about as the behaviorally identified commitment. What causes changes in chromatin accessibility? Well, it's not supernatural. It's driven by transcription factors. And so for many years, my lab has been interested in what are the transcription factor expression changes that are responsible for this highly predictable change in cell potential. And I would emphasize that the reason that I give these stages different names is because we can distinguish these stages by cell surface phenotype, and we can sort the cells preparatively by flow cytometry, and we can show that the cells with this phenotype are not committed and the cells with this phenotype are. And using that information, we can ask which transcription factors change expression in the cells that we sort according to these same characteristics. And there are about 400 regulatory gene loci that change expression to a significant extent across the span of these stages. But some of the most dramatic changes actually occur right in this interval. And these are some key players in the process that I just want to introduce to you at this point, because we're going to refer to them later on. First, the cells come into the thymus as anything but a tabula rasa. They're very healthy, sort of self-sustaining, multipotent progenitors, and they express both genes which are required throughout stem cell development and T cell development, like E protein genes and RUNX genes, and also genes that are used in the progenitors but are not used in mature T cells. And that includes factors like PU.1, which is a famous myeloid cell, very potent lineage regulator. And there are also a whole set of other transcription factor coding genes that I'll tell you about a little bit later in this talk, which have a similar expression pattern, strongly expressed when the cells come into the thymus, extinguished during the commitment process. So this says that the loss of access to these alternative states is correlated with a loss of certain regulatory gene expression. At the same time, we obviously are gaining T cell differentiation status, and so that two of the first transcription factors that are turned on as part of the T cell program are GATA3 and TCF1. They're turned on by the notch signaling, but they're turned on fairly early after the cells get into the thymus. And we know from measuring this many different ways that this upregulation occurs days and many cell divisions before the commitment event occurs. So although this may be necessary, it's not sufficient for commitment. And so now we can ask what is closer linked to the commitment event itself. And the most closely linked gene to this commitment process itself is the gene that encodes this other transcription factor, BCL11B. It's a terrible name for this poor gene, but I'm going to say it many times, so I apologize. It actually is it's a misnomer. It's named for a B-cell leukemia 11A, which is its paralog, but BCL11A is a proto-oncogene and BCL11B is a tumor suppressor. So it just shows you don't go by names all the time. Okay, so when I say that the onset of BCL11B expression coincides the most closely with commitment, this was at first a bulk population analysis correlation that was made, but then we've been able to study this in more depth. A very wonderful then postdoc in my lab, Hao-Yuan Kuei, who's now a faculty member at University of Washington, knocked in a nuclearly localized yellow fluorescent protein gene into the 3' untranslated region of this gene so that the same BCL11B allele could simultaneously encode normal BCL11B protein and this beautiful yellow fluorescent protein that would go to the nucleus. And so we could very beautifully follow whether a cell had turned on that allele of BCL11B or not. And using this as an additional criteria, together with the cell surface markers, additional criterion, we can show a very, very tight correlation indeed. So this is just an example of the cell surface marker profile that we use to track the progression of the cells through these stages. This is the earliest stage, ETP, then DN2A, DN2B, DN3A, DN4. This is the stage when the cells become committed at the bulk population level. But if you then subdivide these populations according to their expression of this BCL11B reporter, the cells down here are the earliest cells. They don't express BCL11B at all. That's the same as background. All of the cells from here to later stages uniformly express high levels of BCL11B. Only in this tiny little DN2A sliver do you have this bimodality. If you sort the cells according to the bimodality, the BCL11B negative cells still are not committed. The BCL11B positive cells are already committed. You can test them, they can't give rise to anything else. And we know that when BCL11B is mutated, the cells can't go through commitment properly and so forth. So we know that it's functionally important for this process. And one large line of research in my lab is studying how BCL11B participates in commitment. I won't talk about that today, but I would be happy to ask you to answer questions if you're interested later on. So, considering how the commitment event is timed, we know that part of it, the timing, is based on AND logic constraints. We know that BCL11B needs at least 4 different positive regulatory inputs, which include the notch signal itself, which is this black arrow, which include the TCF and the GATA3 inputs, which are turned on in a feed-forward network circuit from the notch, and that's the blue and the purple here. And we also know that it needs RUNX, which is there all the time, but which only gains access to the BCL11B locus as a result of a chromatin opening event, which is a very interesting story in itself, at the correct time for BCL11B to go on. But this process actually is completed in the DN2A stage, partway through the DN2A stage, and that's when you see BCL11B come on. But aspects of this system have to be set up much earlier. And the part of what has to be set up earlier is the TCF and GATA3 activation. But part of it also turns out to be the initiation of this slow chromatin opening mechanism, which we've been able to identify based on putting different colored reporters in the 2 alleles and showing that there's actually a constraint. So the 2 alleles in the same cell don't even go on at the same time. And we've been able to solve the modeling equations for this process, which extrapolate the chromatin opening process to a beginning before the cell, significantly before the gene comes on. So something is happening before the gene comes on. And in order to find out what it is, we have to look back at the earliest stage when the cells enter the thymus and try to break that stage apart and see what's going on there. Now that's actually not trivial. That's this ETP population. But I call it the dark phase of T cell development, and I think someone else made a joke about dark phases yesterday. It's a dark phase to the extent that the cells really are not yet expressing any markers that distinguish them definitively as being T cells. And what that means is that although you know that they're not T cells yet, with great certainty, you cannot be sure just by looking at these cells that they're not a mixture of cells of completely different types. So when you look at the genes that are expressed in that population, the burden of proof is upon you to say that those genes are being expressed in the same cells that are going to give rise to the T cells. And so the population gene expression resembles multipotent progenitor population gene expression, but one really has to ask at the single cell level, what is the set of properties that go along with the first cells that are really in the T cell pathway? And this is important because it really addresses this question of how much the hematopoietic pluripotency genes are themselves playing a role in the T cell development initiation, either as an enabling function or as a kinetic constraint on that process. And so obviously we had to do some single cell analysis of this compartment. Now, most people, this has now gotten to be an almost knee-jerk response for biologists who have the money to do this. You don't know what is going on in the system. Oh, do single cell. Well, in this case, we actually knew quite a lot about this system, but we still had questions that we could not answer without the single cell analysis. And so the real question was, how complex is this ETP compartment? We know from population dynamics measurements in several systems that the cells can take as long as 10 days to traverse this compartment, between 1 and 2 cell divisions a day. So that's quite a significant amount of developmental action that could be going on at that period. And we know that the cells are receiving these notch signals the whole time. How many different stages are there within that compartment? Are the cells just in a waiting antechamber, or are they actually moving forward in a programmed way? And so we basically sorted the cells across this continuum, including all the stages that we thought might be going towards the committed state, with the committed state of BCL11B upregulation as an endpoint. And then we subjected the cells to several kinds of analysis. Now, the standard thing to do, the most convenient, is to throw them into a 10x Genomics Chromium single cell RNA sequencing machine to do droplet-based sequencing. And then you get tens of thousands of cells that you can analyze, and you get some sequence information about all of them. It's very, very useful. The sequence comes from all over the genome. There's one big problem with it, though, which is that it sequences at very shallow depth unless you're a multi-millionaire. And the trouble with shallow depth is it means that you have pretty high confidence about the expression status of genes that code for kind of the business molecules of a cell, like cytoskeletal molecules. I'm sure that these cells really express actin. But if you want to know about transcription factors, whose transcripts are often expressed at only 10 or maybe 20 per cell, you have an unbelievably high false negative rate. And that means that you can't confidently say when the cells are turning on the T cell regulatory program. Now, here's a very strong hypothesis. This is not hypothesis free. This is not unbiased in that sense, because we're interested in causal mechanisms. And we know that to get developmental change, you need change in transcription factor gene expression and activity. Therefore, we care deeply about when those transcription factors actually undergo different changes in expression. And so it's not sufficient for us to look at gene expression if we can't say something about the transcription factors. And therefore, we've actually combined this with several other techniques, including a very small cell number but deep sequencing of single cell transcriptomes, and seq-fish, which is like this is in collaboration with Long Kai at Caltech. It's a very similar technique. It's called MERFISH in Xiaowei Zhuang's lab. And essentially what it is, it's a very, very highly multiplexed single molecule fluorescence in situ hybridization method that allows you to count the individual molecules of messenger RNA in a given cell. And I first want to tell you about the seqFISH because it allows us to tell which cells are and are not even eligible to be considered for being part of the T cell program. Now, we first wanted to find out what the subpopulations were. We wanted to order them, but we had to find the regulatory genes then that were active and relevant in the T cell precursors, and then discover what is the model for the sequence of changes in expression of those genes. That's the pseudotime model. We wish to validate the expression patterns of these genes and the order of these states, these cellular states. And then use these as a basis for hypothesizing control functions that might be involved in controlling the progression of cells through these states, which we could then test experimentally. And so I'll try to get through this quickly. The goal for looking at the RNA, at the single cell transcriptome analysis, was first then to look for the genes which are specifically expressed in these early stages, and use them. I don't know if you can see this. I can't see it very well here. But first to find out what fraction of the cells in this early population actually show T cell precursor activity in clonal development assays. So are we talking about looking for a minority fraction, or are we talking about looking at a majority? Asking how, basically, whether the stem and progenitor genes, which are unique to this population, are they in fact co-expressed at all with genes that we recognize as being associated with the beginning of the T cell program? If they're not, it's not clear that they're actually coming from the same cells. And then use them to try to determine whether we actually have all of the cells apparently associated within the same program, or whether we have outgroups that appear to be disjunct from those cells. And then use these to then test whether any outgroups that we see are really T cell progenitors or whether they're unrelated. Then to find out for the cells which are not outgroups, do the gene expression dynamics form a single trajectory, or do they appear to be branched? And then use a number of tests, both bioinformatic tests and cellular tests, to take the model that's made from this computationally oriented set of gene expression states, and ask whether the pseudotime model is correct, by first asking whether we have whether the actual pseudotime model fits the effects that we can measure by short acute perturbation experiments. That is, if we showed this transcription factor expressed before these other genes are expressed, and another transcription factor expressed after those genes go off, do the effects of perturbing those transcription factors appear to be consistent with what we're seeing in this model? The other thing is to look at computational modeling of using velocity calculations, which is a way of using the information from reads of intronic RNA, which may be newly transcribed, versus mature messenger RNA, to ask whether the cell is currently making transcripts from the same genes that it's already expressing, or whether it's actually turning on new genes. And finally, really to ask whether what the cells are saying, and I'm going to get to this in a moment. This is the result that we've seen, and asking whether this is in fact correct by using single cell sorting of the cells according to the genes which come up as putative population separators from the pseudotime model, and then actually testing whether these cells are in fact in the correct developmental sequence. So the first issue is to ask whether the progenitor-associated regulatory genes are actually expressed in the cells that are starting the T cell program. And the earliest known T cell genes that are involved in this are TCF7, which codes for the TCF1 transcription factor, and GATA3. TCF7 is expressed at a reasonably high level, but even so, it's expressed at approximately 50, give or take 50 copies per cell. So it can go up to 150 and maybe down to 20. But even so, it's called as being negative in about 40% of the cells if you use 10x. Therefore, it's very important not to use 10x to determine which cells are and are not expressing this. And the single molecule fish correctly shows that virtually all the cells are expressing TCF7 and RUNX1 and Notch1, and that's consistent with the genetic evidence that, in fact, cells of these early stages are not even viable unless they have these genes expressed. And then we can actually measure the population distributions of transcripts for a whole set of genes, including some that turn on late, like BCL11B, which is off in the ETPs mostly, and then turns on later, and other genes which are already on in the ETPs, but then get turned on more highly as the cells cross to the next stage, which is called DN2. And what these seq-fish messenger RNA counting experiments show us is essentially that GATA3 and TCF are turned on at some low level in virtually all of the ETPs, and then they get much more highly expressed in individual cells as they move into the DN2 state. BCL11B is essentially off in the ETPs, but it comes roaring on in the DN2 cells. All the cells that turn on BCL11B have already been expressing TCF7, so they're all TCF7 positive, and these are where they're turning on BCL11B. And so these are our positive controls, and we can use this approach to ask whether the cells that are in the T cell program, either expressing the early genes, TCF and GATA3, or the slightly later gene, BCL11B, are they or are they not expressing progenitor genes in the same cells? And here's a very good progenitor gene, LMO2, which is associated with hematopoietic stem cells and multipotent precursors. And I think you can see that of this many cells that have turned on TCF7 are still expressing LMO2. Here, the dark blue ones are the ones that are still ETPs. The light green ones are the ones that have already become DN2. Many of the DN2 cells have turned off LMO2, but of these cells, the cells that have turned on TCF are also expressing LMO2. So these can be co-expressed in the same cell. Same for GATA3. Practically everybody who turns on GATA3 in the ETP stage is still expressing LMO2. Whereas if you look at the cells that turn on BCL11B, which happens later in the DN2 stage, these are all light green, which means they're all already DN2, they are not expressing LMO2. So LMO2 goes off between the time that these genes are turned on and this gene is turned on, and but it is definitely on in many of, if not all, of the cells that first turn on TCF and GATA3. So those are the cells that start the T cell program. Now, using this confidence to say that the cells in the T cell program that really start the T cell program are also expressing progenitor genes, we can now use lowered sensitivity data like 10x to get a bigger picture of the whole transcriptome in these cells. And what this is using a display which allows us to lay out the phenotypes of individual cells in a way that does not maximize the dispersion of the cells, but rather puts them into an orientation that brings the most related cells closer together and the most unrelated states further apart across axes that actually make biological sense. This is a force-directed graph, and we're using Alon Klein's Spring software. And what this shows us is that there are actually two almost orthogonal processes going on in these cells, that these cells change gene expression as they develop from most immature at this edge of the distribution to the most mature, which is at this end. So this is least mature. The cells down here are most mature. These are most mature states. But at the same time as the cells go from least mature, a little more mature, a little more mature, a little more mature, there is also a cell cycle axis, and that the cells go from G1 at this end to G2 plus M at this end. And so you essentially have two orthogonal things going on, and that's consistent with the fact that the cells are going through multiple rounds of cell division, even as they develop. But you can also see that not all the genes fall into this great big blob. This is sort of the cell cycle axis. These are the early versus late developmental axes. But here's a little spur here, which seems to be sticking out away from the rest of the world. And it turns out that this population of cells has gene expression properties which are somewhat like progenitor cells, but they're also somewhat like granulocytes, which is a different cell type. We can use the whole transcriptome information in this analysis to purify these cells out and test them for their developmental potential. Lo and behold, they have nothing to do with T cell precursors. They're basically contaminating early granulocyte precursors. And so it's very important to exclude them from the rest of the analysis, because otherwise they screw up the computational analysis of the phenotypes. So using that kind of information and excluding the cells that aren't part of the larger group, we can then actually make a model of what's the order of different gene expression stages in this process. And here we're using software called Monocle in order to organize these states. This was invented by Cole Trapnell. We're basically using other people's software for most of this. And what I'm plotting here this is just a meaningless plot of the order of the states, but this is actually breaking up and mapping onto this the expression patterns of about 763 different genes that are changing the most during this period, which allow us to use them as landmarks to see what's happening. And why am I showing you this? Because there's one very simple model that we had going into this, which is that the cells come in with a stem and progenitor gene expression program, which gets turned off in the process, and they come in not expressing T cell genes. 5 minutes? You're serious? Okay. Okay. I'll skip my talk then. I won't give my talk, but I will tell you that we found some interesting things out about this. Okay. So I will skip the fact that we've actually validated this, and that we actually know that we're correct. And so this is, if any of you are interested in real validation experiments, I can take you through it. This is all recently published. So if anyone's interested in looking this up, you can actually see what a validation experiment looks like. And it's done. Where I want to go with this is to say that you actually get something out of this, which is the fact that you can use this information then to ask about regulation and to look for new regulators of the process. And so the question, as I told you, there's a whole set of these genes which come in being expressed in the progenitors but are turned off in the process. But we still have to explain why the process takes so long, and why in the slides that I skipped over, we did not see a simple turning off, turning on, but we actually saw several intermediate waves of change, which suggests that the process that the cells go through is actually more complicated and more multi-state than a simple turning off, turning on. And so the question is, which of these transcription factors in this set, with somewhat different times of being turned off, could be playing some kind of a role. And essentially what we can do, there is a validation experiment. There's actually an assay you can do to test this. And this is an attempt to get at causality. What we can do is, normally the T cell developmental process is irreversible. But the irreversibility includes turning off a whole bunch of regulators. And so the question is, how much of an impact can we have on the phenotype of the cells if we add them back again to a cell that's already finished that process and which is fully expressing the commitment-specific genes? And so if we add back any of these earlier genes, do we actually get the cells to go backwards? And the first candidates that we studied were based on molecular evidence and some hematopoietic functional evidence, which the gene PU.1 or SPI1 encodes this master regulator of myeloid cell potential. Myeloid cell potential is something the cells have when they come in. It's something they lose during commitment. Loss of PU1 should make sense for that. PU1 also turns out in more recent work that we've published over the last couple of years, it's the main transcription factor binding to the chromatin sites that are specifically open in those early stages. And it works as a pioneer if you add it back and so on like that. The problem with PU1 is that it does not really strongly activate the set of other transcription factor coding genes of the pre-commitment state. So although it has an effect, and although it acts as a great placeholder for the pre-commitment open regulatory elements, it doesn't in itself activate the program backwards. And so if I have a chance to show you a couple more slides, I will show you what does. And that is a very interesting complex because it's actually a biochemical complex made of polypeptides that are encoded by 3 different genes, which have actually different patterns of expression. One of them, E2A, is expressed at an absolutely unchanging rate all the way throughout. And so you would assume it has absolutely no developmental role at all. It's just there as background music. But it actually turns out to be important because it heterodimerizes with this other chain called encoded by the LYL1 gene. They form a DNA binding heterodimer, which actually is a little different than what E2A would be in terms of its DNA binding preference when it's expressed by itself. And the real game changer is this gene that I showed you a little bit about before, LMO2, which is expressed very early. And it encodes a non-DNA binding subunit, but one that's very important for the function of this DNA binding heteroduplex, because it actually stabilizes this heteroduplex and it gives it new chromatin opening functionality. And what the point I would like to make is that this complex, which is rate limited by the availability of LMO2, turns out to be incredibly potent at determining what the cells will do. So we can take cells that have already differentiated past the point when they've turned off LMO2, and which they're in the process of turning off LYL1. They have E2A, but that's just always there. And we can test them by adding LMO2 back to them. And we can then take LMO2, put it into the cells after they've differentiated past this point, and then put the cells back in culture, let them differentiate more, and ask, do they stay committed or do they do something else? And the answer is that they completely reverse differentiation. We've never seen anything as powerful as this before. Essentially, all the genes which are the hallmarks of the uncommitted state, which had been off in these cells, they come roaring back on again, while most of the genes which are specific for the committed state get downregulated as a result of this very quickly. We have a massive effect of this single complex on chromatin openness. And in effect, in one shot, this regulatory factor complex has reactivated the entire early network while downregulating the other network. We can do, because of the accessibility of this system, which I haven't had a chance to tell you about, it's a beautiful system for being able to perturb this in real time and watch exactly how the cells over the next 36 or 48 or 72 hours react in real developmental conditions. And we can also then relate this to the opening of specific chromatin sites, the closing of others. We can also ask how much of this effect is due to the direct impact of LMO2, LYL1, E2A, and how much is due to the other genes that they activate by doing a combined gain and loss of function perturbation using gain of function plus Cas9 CRISPR deletion. And so we know a lot more about that, which I won't have a chance to tell you about. But the point is that the cells will even reverse their BCL11B expression. So this is just using the BCL11B fluorescent reporter cells. These would be the control cells which would go over this period to turn on BCL11B. If you put this LMO2 in when the BCL11B is already turning on, basically it squishes it back down again, it turns it back off again. So the cells are really gone back, and they get back alternative developmental potentials. So essentially if you get the right factor, you can actually hit the apex of this gene regulatory network, and you can reactivate the entire progenitor state in these cells. And this is physiologically relevant because these states in fact correspond to the real single cell phenotypes that we can see in the genuine intrathymic precursors of these early intermediates. So the conclusion is that essentially the differentiation of these cells is actually slowed and actually made somewhat stochastically noisy in terms of time, partly because there's complex gene network dynamics where you have a self-reinforcing progenitor state which can renew itself over the first 10 odd cell divisions while the cells are in the thymus, but which is being opposed by an accumulation of more and more of these notch activated T cell transcription factors. We can actually relate this to a pseudotimed trajectory that I didn't have a chance to tell you about in detail. Okay, let's go back to the conclusions. That we can actually identify at least one extremely important regulator that has the power to cross-activate its own gene regulatory circuit partners to establish a really strong state of progenitor status, and make development go backwards. And it means that the developmental reversal is sensitive to single gene expression, and that basically this has a very interesting consequence that even the earliest expressed transcription factors may ultimately have a legacy behind them, that their own timing of silencing may set the clock for all the subsequent events that allow the cells to continue their T cell development. So if you're interested, I'd be delighted to answer more questions about this. I just jumped over the details, but I just wanted to give you a feeling for what we've worked on. The single cell analysis was done by Wen Zhou in my lab in collaboration with the Barbara Wold lab and the Long Kai lab. Shun Wang did all of the LMO2 story. Mary Yui is the main person in my lab who really discovered commitment and who's mentored everybody younger in my lab about the cell biology of the system. We've had wonderful collaborators inside and outside the lab, and if my chairman will let me, I'd be thrilled to take questions, and if not, I'll be happy to talk to you guys later. Thank you very much.
For question?
Ellen, this is absolutely fascinating. So is that a meaningful question to ask, whether this differentiation has to do with disappearance of activators or appearance of depressors, or this is too simplistic?
I love this question. Thank you.
Because most of your arrows were activating arrows.
Yeah.
With a few exceptions.
Yeah. So we've burrowed into a couple of these transcription factors with some seriousness, and we've worked very hard on PU1. We've worked very hard on BCL11B itself, and we're working with increasing intensity on LMO2, LYL1 as a complex. When you compare the actual sites of binding with the sites that open and with the status of the local gene expression, what you see is that each of these factors tells a different story. BCL11B is preferentially a repressor, but it can participate in activating complexes. PU1 is mostly an activator if you look at where it works directly, but in fact it has a very cute method of working as a repressor indirectly. It's so powerful at nucleating the binding of other factors like RUNCs, which are like currency for the system. It can pull RUNCs away from places where RUNCs is acting as a positive regulator with other factors, and therefore starve those cells of the starve those genes of RUNC's input. So that's an indirect method. LMO2 seems to be mostly positive, but it activates at least one fairly dedicated repressor. So we think that there's going to be a lot of functional complexity, and we're working on this very hard. Thank you.
Would you predict that the LMO2-LYL complex, if ectopically expressed, is strongly leukemogenic, given its ability to block end-stage differentiation?
Yes, so LMO2 is famous as a proto-oncogene, and LMO2 expression later in T cell development, artificially, is associated with a slow progression towards leukemia. Under those conditions, those experiments were done with transgenics that express LMO2 essentially after LYL1 is mostly gone. So there's LMO2 itself doesn't bind DNA, so the only cells that can become leukemogenic there would be the last few that are still expressing LYL1. In our situation, everybody expresses LYL1, or we add it back, and then it's an immediate effect. It's not slow at all. These cells are not becoming leukemogenic in our system because they're too early and they don't have the signaling activated through the T cell receptor. So what's happening in the leukemias where LMO2 is famous is that it's being expressed with a little bit of late LYL1 into the stages when the cells are getting this massive RAS-MAP kinase activation through T cell receptor, and that combination is apparently leukemogenic, but it takes a while, and so we think there are other events involved, which will be, we'd love to talk with you about more, because I think there are many other mutations that probably occur before the leukemia really blossoms out.
Alan, thank you.
Thank you.
Two short questions. First, how is the opening of the chromatin controlled? And second, I understand you used mathematical modeling, and I know that you sometimes get quite unhappy about it, but you wouldn't tell it as you. But these mathematical models, they probably don't give you a causal analysis. And so how do you deduct from there or from wherever your causal question?
Yeah, well, I had to cut all those slides, unfortunately, I'm really sorry about that. I didn't realize how much time was going by. The causal analysis is all experimental. And so it all involves taking and I can just show you the slide without explaining it. We can basically say that the pseudotime is correct by sorting the cells according to their position in the pseudotime and actually doing a clonal efficiency and developmental kinetics analysis and correlating that with clonal transcriptomes. So for the perturbation, for the causality, we're actually taking the cells in one state, which we are documenting. We're adding something or subtracting something, and we're measuring the descendants of those cells 2 or 3 days later and seeing how they changed, comparing them with controls. So we are actually inducing under highly controlled synchronous conditions the change, and we're doing it with one set of perturbations which are not mimicked by another. So our causality is based on empirical testing. So there is causality in there. The problem is to come up with the correct mathematical model to explain how many component processes there are in there and what the dose response relationships actually are. And the frustration has been that the system, we essentially have to, it's one of these crimes that Dan was talking about yesterday. We have to put everything we know into the system and then fit the model to everything we know, and it gives us back everything we know, but that's fake, so that's circularity. And so we're not happy with that. That's not actually giving us true predictiveness.
How far can you go without measuring protein concentrations?
We can't measure protein concentrations on the 10 to the 3rd to 10 to the 4th cell level, or the single cell level in any meaningful way. So I think we're forced to make do with the RNA, and we are forced to make do with the RNA and the attack sites. And we're forced to make do with that, knowing that the cells are proliferating, and therefore that if the expression isn't sustained, it will wash out.
Unfortunately,
Thank
we
you.
have no more time for questions, so we can continue the discussion after the break. So let's thank Professor
Thank you.
Rothenberg.
Thank you.